Today we'll be talking about the Hardy-Weinberg equilibrium. This type of equilibrium occurs when genetic frequencies in a population do not change over time. In order for a population to be in this equilibrium, there must be no migration, no mutation, no natural selection, completely random mating, and an infinitely large population. If all these assumptions are met, then a population will be in Hardy-Weinberg equilibrium after one generation of random mating. So, how can we determine that a population is in this equilibrium? Let's take a look at a sample population of penguins living in Antarctica. We'll be focusing on the gene that determines the shape of the penguin's beak. We have two alleles, big H and little h. Big H results in a hooked beak, big H, little h results in a thick, medium-sized beak, and little h results in a small, thin beak. Well, the Hardy-Weinberg principle also tells us that if a population is in equilibrium, it will fit into the following equation. In this equation, P is the frequency of one allele, and Q is the frequency of the other allele. Let's say that the big H has a frequency of 0.7 and the little h allele has a frequency of 0.3. We can plug in the frequency of our big H as our P value and the frequency of our little h as our Q value and see if the equation is balanced. We will square our P and our Q, which will give us the genotypic frequencies of our homozygous genotypes, and then multiply PQ by 2, which will give us the frequency of our heterozygous genotype. Adding all of those up, we can see that our sum is 1, meaning that our population is indeed in Hardy-Weinberg equilibrium. But what happens when one of the assumptions of the Hardy-Weinberg equilibrium is violated through something like natural selection? Let's take a look. These are the fitness levels of the penguins on this island. We can clearly see that the hooked beaks have a higher fitness than the small, thin beaks because the hooked beaks are more beneficial when trying to catch fish. This creates a directional change towards the homozygous dominant genotype. What will happen to our genotypic frequencies in the next generation? We can actually figure this out by first multiplying the original genotypic frequencies by our fitness and then dividing by the average fitness. We already have our original frequencies from our equation earlier as well as our fitness, so all we have to do is calculate average fitness, which is a weighted average, and we can do this using the equation shown below. Essentially, we will be multiplying the genotypic frequencies by each fitness and then adding all those numbers up to find the average fitness. In this case, the average fitness is 0.8635. Now we have everything needed to calculate the genotypic frequencies of the next generation. We have already multiplied the initial frequencies of each of our genotypes by our fitness. We can then divide that number by the average fitness to determine the relative genotypic frequencies after selection. In this population, we can see that the frequency of the homozygous dominant genotype increases and the homozygous recessive decreases as selection favors the hooked end of the spectrum. After infinite generations, we will eventually reach a completely homozygous dominant population.